Hi students, good morning to everybody. I would like to continue the lesson Body Fluids and Circulation. Today's topic is the blood vessels. What are the types of blood vessels? The arteries, veins and capillaries. So the blood vessels carrying blood are of three types. Number one, arteries. Number two, veins. And number three, capillaries. Now these blood vessels are hollow tube-like structures and have complex walls surrounding the lumen or the cavity. They have complex walls made up of various layers surrounding the lumen. Complex walls surrounding the lumen. There is a cavity. So the blood vessels are hollow structures and have complex walls because they are made up of many layers surrounding the lumen. If you are taking the wall of the blood vessels in humans are composed of three layers. The blood vessels are composed of three layers, namely tunica intima, the innermost layer, then tunica media, the middle layer, and tunica externa, the innermost layer. The tunica intima is also called tunica interna, and the tunica externa is also called tunica adventitia. We'll see the structure and diagram too. As I mentioned earlier, the walls of blood vessels, either the arteries or veins, are made up of three layers. Number one, tunica interna or tunica intima. It is an inner layer forming what is called the basement membrane that supports the vascular endothelium. The inner layer formed a basement membrane on which we have the squamous epithelial cells are formed. Those squamous epithelial cells lining the blood vessels are called endothelial cells. This is nothing but squamous epithelium. As it lines the blood vessels, also the lymphatic vessels, it is called endothelium, nothing but squamous epithelium. Then tunica media, the middle layer. It is made up of what is called actually the smooth muscles and an extracellular matrix. Matrix means a ground substance. Okay, so it is a middle layer composed of number one, smooth muscles, and number two, extracellular matrix, meaning outside the cell. Matrix, a ground substance. That matrix contains a protein, what is called elastin, made up of elastic fibers, highly elastic in nature, hence the name elastin, the name of the protein elastin. So the muscle fibers along with elastin responsible for the contraction and relaxation of the muscles, which result in vasoconstriction and vasodilation. Vasoconstriction and vasodilation. Constriction means decrease in diameter. Dilation means increase in diameter. So the stretching of, the increase in diameter of, decrease in diameter of the blood vessels are brought out by the contraction and relaxation of the smooth muscles along with the elastic fibers made up of elastic protein. Now, tunica externa or tunica adventitia. Two names, tunica externa, tunica adventitia. So interna, inner, externa, outside. Tunic means covering, hence the name tunica. Now it is an outer layer composed of connective tissue, which is rich in collagen fibers, a kind of protein. Collagen is a kind of protein, a fibrous protein. So the tunica adventitia, the outermost layer, is formed of connective tissue with collagen fibers. These are the simple diagrams showing the cross section of an artery and a vein to show the various layers of the wall. Either in the case of vein or in the case of artery, we have four layers. Normally we can say three layers. Now endothelium plus tunica interna just together form tunica interna one. Now the outermost layer is called tunica externa, made up of fibrous connective tissue rich in collagen fibers. The middle layer is tunica media made up of smooth muscles, along with extracellular matrix with the elastic fibers made up of elastin protein. If you compare the muscular layer, the tunica media, it is thick in the case of artery and thin in the case of vein. So I represent that vein. Thin in the case of vein and thick in the case of artery. The innermost layer is the tunica interna, which is supposed what we have a single layer of squamous epithelium called endothelium. Now the black layer represents what's called tunica interna, forming the basement membrane on which the cells are arranged. What we have the endothelium. The innermost layer, single layer of what we have endothelial cells, nothing but squamous epithelium lining in the wall. If you compare the lumen, this is a cavity of the lumen. The lumen is narrow in the case of artery. The lumen is wide or long in the case of vein. This is because of difference in thickness of the muscle layer. The muscle layer is very thick in artery, that's why the lumen is narrow. And in the case of vein, the muscle layer is thin, that's why it's normally, that one, the lumen is wide. Now let's see what are the characteristics of arteries. Those blood vessels that are leaving from the heart, that is carrying blood away from the heart, they are called arteries. Any blood vessel. Normally we can see the arteries are those blood vessels which can carry oxygenated blood. Any blood vessel leaving from the heart, whether it carries oxygenated blood or deoxygenated blood, they are called as arteries. For example, we have aorta that carries oxygenated blood. The pulmonary artery that carries deoxygenated blood. So anyway, any blood vessel leaving from the heart, they are called as arteries, irrespective of the blood they contain. They carry either oxygenated blood or deoxygenated blood. Then, so regarding the position, the arteries lie deep inside the body. They are deep inside the body. And this one away from the heart. If you are comparing the walls of arteries of veins and then the walls of arteries and veins, the walls of the arteries are thick normally and non-collapsible because they have to withstand high pressure, the blood pressure. We are calculating the blood pressure only in the arteries. Due to the pumping action of the heart, we have the pressure is always high in the arteries. So to withstand the high pressure, the walls of the arteries are thick in nature and also non-collapsible. The thickness is due to the thickness of what is called the theca media, the muscle layer. Now usually the walls are absent, no walls, unlike the veins. The veins have semilunar walls, but the walls are absent in the case of arteries. I already shown in the diagram, the lumen of the artery is narrower than that of the veins. This is because of increased thickness of what we have, the media, what is called theca media, the middle layer. Then all the arteries carry oxygenated blood, but there is an exception. That is the pulmonary artery. The pulmonary artery, which makes its origin from the right ventricle, carries impure blood, we can say deoxygenated blood, towards the lungs for purification. Excepting that one, all of the arteries carry oxygenated blood. Now the large artery in the body is the aorta. The large artery in the body is aorta. If we are making the diameter of that artery 2.5 centimeter in diameter and 2 millimeter in thick. Now this artery, aorta, branches into smaller arteries. They just normally see that one culminate in just into the tissues. And normally they culminate into the tissues as what we have somehow smaller arterioles, and these arterioles are called feed arteries. I'm using the word arterioles, but in the book it is given feed arteries. So the large artery, the aorta, branch into smaller arteries and they culminate into the tissues, and in the tissues as feeding arteries because they feed the cells with what we have, the nutrients and oxygen. Okay, that's about what we have. Now if we are comparing the pressure in different blood vessels, in the arterioles, the BP is equal to 85 millimeter mercury. In terms of SI unit, in SI unit, the millimeter mercury is what is called kilopascal. So in arterioles, we have the BP is 85 millimeter mercury equivalent to 11.3 kilopascals. Whereas in the case of capillaries, the BP falls down, the drops of what is called BP in the capillaries. It's about 4.7 kilopascals equivalent to 35 millimeter mercury, because 1 millimeter mercury is equal to, I write down on the board, 0.13 kilopascal. So SI unit of 1 millimeter mercury is equal to kilopascal. That is, actually it is expressed in terms of kilopascal. That is the SI unit of 1 millimeter mercury, or millimeter mercury is expressed as kilopascal. Okay. If we are comparing the blood pressure in different, just the branches, either in the case of artery or arteriole or capillaries, the blood pressure is always high in the case of arteries, for example, the aorta. And about the arteriole, the blood pressure is about, that is 85 millimeter mercury. That is equivalent to 11.3 kilopascal. So 1 millimeter mercury is equal to 0.13 kilopascal. It is nothing but the SI unit of millimeter mercury. So the SI unit of millimeter mercury is kilopascal. So it is equal to 11.3 kilopascal in terms of SI unit. So 85 millimeter mercury in the case of arteriole. If we are comparing the pressure in the case of capillaries, the capillaries normally the pressure drops down, and it is about 35 millimeter mercury equivalent to 4.7 kilopascal in SI unit. Now 1 millimeter mercury is equal to 0.13 kilopascal. So SI unit of millimeter mercury is kilopascal. That's what I mentioned earlier. Okay. So arterioles, these are the branches of arteries. These are thin-walled, small vessels which are connected to the last part, what we have the capillaries. So we have artery, arterioles, the last branch, the arterioles, which are forming a bed responsible for exchange of various materials between the blood and what we have the tissues. So at the junction of the arteriole and the capillary, we have a small sphincter that regulates the flow of blood between what we have the arteriole and the capillary network. So a small sphincter is present at the junction of what we have the arteriole and the capillary network to regulate the blood flow. Now, normally in some cases, normally you know the arteries divide into arterioles, the arterioles into the capillaries. In some cases, the arteries do not branch into what we have arterioles and then capillaries. There is no division, there is no branching of the artery into arterioles. Instead, they form anastomosis. What are anastomosis? So, anastomosis are connections between two blood vessels, connecting two blood vessels. Suppose this is a normal blood vessel, an artery. Sometimes if this artery is blocked, we have a bifurcation. The bifurcation is called the anastomosis. So this is a block A. Now this is nothing but the connection between one blood vessel to another blood vessel. And this is what is called anastomosis. It occurs only when there is a block in what is called in the artery concerned. So it allows the free flow of blood when the artery is blocked, the normal artery. So these are connections connecting one blood vessel, namely the artery, with another artery when the artery is blocked, and this is called anastomosis. So normally the artery do not form, or we have the branches always. Sometimes because of the blocking of the arteries, we have anastomosis are formed. These anastomoses are nothing but the connections connecting what we have one artery to another artery, or one blood vessel to another blood vessel. So normally, if you are taking for example the knee joint, we have a number of anastomoses. These anastomoses actually just when one blood vessel closes, they just allow the free flow of blood, for example while bending the knees. So while bending the knees, there is a chance of closing of one blood vessel. At times we have a bypass, and that is nothing but the anastomosis, transporting or allowing the free flow of blood from one blood vessel to another blood vessel. That's why we have in the knee region or in the joints, we have a number of what is called anastomosis. Now the second type of blood vessel, we have the veins. If you are comparing the arteries, veins have thinner walls. The walls are very thin. And a large lumen. The lumen is large, what is shown in the picture, and hence can be easily stretchable. Then they carry deoxygenated blood except one vein, what is called pulmonary vein. It takes oxygenated blood from the lungs to what is called the left atrium of the heart. So excepting the pulmonary vein, all other veins carry impure blood or deoxygenated blood. Now the blood pressure is low because the blood is returning to the heart. In arteries, the BP is high. This is because of the pumping action of the heart. But in the veins, as the blood is returning back to the heart, only the pumping mechanism brought about by the muscles, the blood pressure is low. So we're always taking blood pressure only in the arteries, not in the veins. It is very low. Now, if you're taking the tunica media made up of smooth muscles, it is thinner than the arteries. That's why it is not actually what is called, it is collapsible. It is easily collapsible. Okay, not withstand high pressure as the muscle layer is very thin with what we call the elastic fibers, also very less. And now what will happen? They can be easily stretched but cannot withstand high pressure. Now, we have the unidirectional flow of blood. Suppose this is a vein, we have the unidirectional flow of blood. So only the blood is moving towards the heart and the backflow of blood is prevented because of the presence of certain walls. These are all the walls. These walls are called semi-lunar walls because they're looking like what is called half moon, semi-lunar walls. So the veins are internally provided with what we have semi-lunar walls. That's why we have the unidirectional flow of blood. From the various parts of the body, the blood is moving or flowing towards only to the heart, not in the back direction. This is because of the presence of semi-lunar walls. They are also called as the pocket walls. Pocket walls. In some books it is given pocket walls. They prevent the backflow of blood. It is one of the important features of what we have the veins, and such walls are absent in the case of what we have the arteries. So they are absent in the case of arteries. Normally, when you want to just find out any defect in the blood samples are taken only from the veins, not in the arteries. Because in the case of veins, the BP or blood pressure is very low, so the bleeding does not occur mostly. But in the case of arteries, as the pressure is high, when you are taking what is called a small opening in the arteries for sucking the blood, automatically bleeding occurs. That's why the blood samples are always taken for test analysis only from the veins, not from the arteries. This is because of low blood pressure in the veins. Okay, this is one of the questions. Why samples are taken? The blood samples are taken from the veins, not from the arteries. This is because of low blood pressure. Okay, I mentioned already the blood samples are taken only from the veins. Why? Because the low pressure in the veins. That's the answer for that one. Now the third type of blood vessels, what we have the capillaries. The artery divides into arterioles. The arterioles divide into capillary network at the arterial end. And likewise we have at the venous end. This is arteries end and the venous end, where we have the capillaries unite together to form venule. The venules are actually joined together to form the vein. Now if you are taking the arteriole and venule, they have tunica, what is called the externa, and endothelium. In both arteriole and venule, we have tunica externa and endothelium. If you are taking capillaries, the capillaries are made up of only endothelium in the form of squamous epithelium. Now, the third one, capillary beds are made up of fine network of capillaries. The capillaries are thin walled and made up of a single layer of squamous epithelium. Okay, suppose you are taking a capillary network. So, capillary like this is a capillary. Is being formed of a single layer of what is called endothelium in the form of squamous epithelium. This is what we call the capillary. This is the capillary being formed of only one layer, a single layer of squamous epithelium, which is called endothelium. Now the tunica media, the elastic fibers are absent. We have only tunica, just actually only the capillary is formed of, that is endothelium. No tunica media, no tunica adventitia. That is externa. In the venule and in the case of arteriole, we have tunica, that is adventitia or externa, along with endothelium. But in the capillaries, only endothelium alone being present. Now, what is the role of these capillary beds? So the capillary beds are the site of exchange of materials between the blood and tissues. So these are all the connectors between the tissues and what we have the blood. So exchange of materials between the blood and what we have the tissues is taking place through the capillary beds. If you are comparing the volume of what is called the fluid in the capillaries, so the blood volume in the capillaries is always high. The blood volume in the capillaries is always high, having high volume. But regarding the flow of blood is normally slow in nature. The flow of blood is normally slow. See that one. So the volume of the blood is normally high and the rate of flow is normally slow in the case of capillaries. So normally the capillaries contain mixed blood, that is oxygenated, deoxygenated. Now the arteriolar capillaries, or the arteries and in capillaries, have oxygenated blood. Then the venules, the capillaries which join together to form the veins, they contain what is called deoxygenated blood. So they contain, we can say, mixed blood, that is deoxygenated and oxygenated blood. As I mentioned earlier, the capillaries contain the mixed blood, oxygenated blood in the capillary of an artery and deoxygenated blood in the capillary of vein. If you are making a section of what is called a venule formed by the union of capillaries, arteriole, the one which is formed by the division of arteries, and capillary. Now in the case of venule and in the case of arteriole, we have two layers, tunica externa and endothelium. Here also tunica externa, endothelium, there is no tunica media. And if you are taking what's the capillary, it contains only endothelium. Even you have, that is tunica adventitia, tunica externa is absent. There's a main difference between these things. In both venule and arteriole, we have tunica externa and endothelium, and in capillary only endothelium alone being present. Okay, that's about the capillaries. A few facts we have to know. What are anastomosis? I explained already. I'll represent in the form of notes. What are anastomosis? I mentioned already. These are connections of one blood vessel with another blood vessel. This is number one. They provide alternate route, just normally a blood flow. Suppose if one of the blood vessels is blocked, if the original blood vessel is blocked, now this is the original blood vessel, there is a block here. So we have alternate route for this blood vessel. So connecting these two arteries, number one, number two. Now this is called anastomose. So normally they provide alternate route of blood flow. If the original blood vessel is blocked, now this is the block. Now we have a number of arteries in the joints to form anastomosis. What is that important? This allows blood flow to flow freely, even if one of the blood vessels closes while bending the joints, for example like this, or bending the knee joints, etc. So during bending of knee joints, so normally we have more anastomosis only in the joints, because while bending, any one of the blood vessels may be closed. At times what will happen, there is an alternative route in the form of anastomosis that just normally allow the flow of blood in the alternative route connecting one blood vessel to another blood vessel, and this is what is called anastomosis.